Good afternoon, everyone. My name is Shikha Singh, and I'm a third-year medical student at the University of Pennsylvania Perelman School of Medicine. And today I'll be presenting on a paper titled Structural and Functional Integration of Human Forebrain Organoids with the Injured Adult Rat Visual System. In this presentation, I'll take us through a brief tour of all of the figures in the paper to give a good overview of the paper's results. This paper was published earlier this year in Cell Stem Cell, and it comes from the lab of Dr. Isaac Chen in the Department of Neurosurgery at Penn. It's been a privilege of mine to be a part of this published work, as well as future endeavors in Dr. Chen's lab. To start off, the paper has 3 highlights. Number 1, it shows that it's possible for brain organoids created from human cells to integrate with the adult rat brain, specifically in the injured visual cortex. Number 2, it demonstrates how organoids can structurally integrate with the host brain through synapses, specifically to the rat retina and visual system. And lastly, number 3, the paper demonstrates how neurons in the organoid can respond functionally to host visual stimulation and adopt feature selectivity as well. The graphical abstract provides a good project overview as well as visual representation of the 3 highlights we just mentioned. In this project, an aspiration cavity is made in the visual cortex of rat brains. An 80-day-old cortical organoid is then immediately transplanted and allowed to integrate with the rat brain for a set period of time, after which the animal is sacrificed and the brain tissue is analyzed for connectivity or structure and function. Connectivity explores both anterograde and retrograde connections from the organoid, while function explores evoked activity and orientation selectivity of organoid neurons. In Figure 1 of the paper, the remarkable survival and development of human cortical organoids following transplantation into the injured visual cortex of adult rats is explored. So firstly, the protocol used for generating these cortical organoids is outlined in Figure 1a, with detailed information available in the STAR method section of the paper. Moving on to Figures 1b and 1c, you can see bright field and fluorescence micrographs of whole organoids captured at different in vitro time points, revealing the presence and growth of multiple cortical units within each organoid. 1D then offers us a closer look through immunofluorescence staining, showcasing the distinctive structure of a cortical unit, which consists of a central lumen with neural progenitors, shown through the PAX6 stain, and differentiated neurons, shown through the MAP2 stain. The neural progenitors are located proximal to the central lumen, while the differentiated cells are found more distal to it. Lastly, Figure 1e demonstrates the development of a rudimentary laminar structure within the organoid, employing markers for upper layer, SAPB2, and lower layer, CTIP2 cortical neurons. So this figure overall provides us key insights into the structure of these human cortical organoids. Now continuing with Figure 1, the focus shifts to the transplantation paradigm of the organoids. So Image F features a schematic representation of the transplantation procedure, where the precise location of the 5mm craniotomy is illustrated. Image G then shows us bright field images of the same animal at 2 months post-transplantation, both under visible and ultraviolet light, highlighting the integration of the GFP-positive organoid within the surrounding brain tissue and vasculature. Image H then shows us coronal sections of animal brains at 1, 2, and 3 months post-transplantation, demonstrating the growth of the organoid over time. This growth is then quantified in Image I, which reveals an expected increase in organoid volume over time, with the red line denoting the average volume at the time of transplantation of these organoids. Now shifting attention to vascularization, images J through L document the development of blood vessels within the transplanted organoid, shown through this CD31 stain. These blood vessels are likely originating from the host brain, as they're GFP negative, compared to the organoid, which is GFP positive. This phenomenon is quantified in Image M, where a statistically significant increase in the number of these distinct CD31 positive vascular structures are found in organoid cross-sections by 3 months post-transplant. Finally, Image N tracks the temporal progression of apoptotic cells within the organoid, shown through the CASP3 stain, revealing a visible decrease from 1 to 3 months post-transplantation, which is shown again in Image O, which quantifies this decrease, emphasizing its statistical significance. This comprehensive analysis sheds light on the dynamic changes that occur within transplanted organoids over time, encompassing their growth, vasculature development, and reduction in cell apoptosis. In Figure 2, a comprehensive panel of stains is presented to elucidate the evolving cellular composition within these organoid grafts, offering us insights into their progressive cellular maturation at the 2-month post-transplant mark. To provide context, image A serves as a low magnification micrograph for the subsequent high magnification images in the rest of the figure. The human origin of the majority of the graft cells is confirmed using the stem 121 stain in image B, while images C and E reveal the presence of the neural progenitors we talked about through the PAX6 and SOX2 staining. Quantitative analysis of the PAX6 positive cells is shown in image D, and it indicates a decline in this population over time. Meanwhile, neuronal differentiation is depicted through the MAP2 and nu-N stains in images F and G, with quantitative data in image H showcasing an increase in the number of mature neurons within the organoid graft. Furthermore, a majority of the graft cells express FOXG1, which is a telencephalic marker, as shown in image I, which is what we expect. Delving more into the cellular identity, images J through N reveal the expression of markers corresponding to cortical layers specifically. So for example, cortical layer 5 shown through TBR1 and CTIP2, cortical layer 4 shown through NECAB1, and cortical layers 2 and 3 shown through CUX1 and SAPB2. Quantitative analysis of these markers is shown in image O, which demonstrates a progressive increase in the number of CTIP positive, CUX1 positive, and SAPB2 positive cells, accompanied by a reduction in the TBR1 positive cells, while the NECAB1 positive cell numbers remain quite stable. Astrocytes, identified by GFAP in image P, exhibited a gradual increase, as shown in image Q, with reconstructions of confocal Z-stacks revealing no colocalization with GFP, indicating that these astrocytes were of a host origin as opposed to an organoid origin. And finally, oligodendrocytes, identified by OLIG2 in image R, display statistically significant increase by the 3-month mark, shown in image S. Now, this analysis shows us the dynamic cellular changes that take place within these organoid grafts over time by looking at neural progenitors, neuronal differentiation, cortical layers, astrocytes, and oligodendrocyte populations over time. Figure 3 then shines a light on the extensive projections that come from the transplanted organoids into the host brain. Figure A shows us the approximate location of these projections at the 2-month post-transplantation mark. Images B and C are low and high magnification views of the host cortex adjacent to the transplanted organoid on a coronal section, showing a high density of the GFP-positive projections in this adjacent cortex. Variations in the GFP border of the graft are also observed. At times, they appear hazy due to the extensive projections into the host brain, such as in Figure 3b, and at other times, they display a distinct border, such as in Figure 3d. These GFP-positive projections also extend to more remote regions, including the retrosplenial cortex in Figure E, the auditory cortex in F, and the visual cortex in G. Moreover, these projections are also identified in the ipsilateral and contralateral corpus callosum, shown in H and I, as well as the thalamus in J, with colocalization of GFP-positive processes with the stem 121 stain we've previously talked about. These projections also show colocalization with NF200 in image K, indicating their dendritic characteristics, as well as with tau, an axonal marker, shown in image L, although these connections don't appear to be myelinated, as they're MBP negative. Continuing in Figure 3, the presence of human synapsin protein at the interface between these graft projections and the host brain suggests the formation of synapses, as in image M. In image N, the number of GFP-positive projections is quantified in different regions of the brain at 1, 2, and 3 months post-transplant, revealing that while the density of graft projections increased in some regions over this time period, the targets themselves remained relatively stable. These projections primarily involve the visual system, which is consistent with previous neural transplantation studies, although we did observe some off-target projections as well, especially in the motor cortex and motor nuclei of the thalamus. Additionally, image O quantifies the GFP-positive projections in different thalamic nuclei at 2 and 3 months post-transplant, with projections observed in nuclei associated with visual and limbic circuits, particularly the LG, the lateral geniculate, and the LP, the lateral posterior. This analysis demonstrated how these transplanted organoids can project extensive GFP-positive extensions into various regions of the host brain, including adjacent and remote cortex areas, the corpus callosum, and the thalamus. While there are some variations in the border of GFP labeling, the projections primarily targeted visual-related regions. Figure 4 unveils the intricate connectivity of these organoid grafts, with a focus on their capacity to receive afferent inputs from various regions of the rodent brain. This dual connectivity that encompasses both afferent and efferent interactions plays a pivotal role in the functional integration of graft neurons into the host visual network. The exploration begins with schematic A that outlines the timeline for employing the modified rabies system for monosynaptic retrograde tracing. Staining of the transplanted organoids with STEM121 confirmed their human origin, as done previously, and it also revealed the presence of abundant DS-red signal in figures 4b to 4d. This shows the sustained expression of the RABV, or the rabies tracing mechanism, in vivo. Cells that were then infected by the virus exhibited both DS-red and GFP, rendering them yellow, while the GFP-positive cells are identified as transsynaptic partners that were located upstream of these starter cells infected by the virus. Now, focusing on the monosynaptic input to the organoid graft, we saw contributions from the adjacent visual cortex, shown in figures 4E to 4G, as well as more distant somatosensory cortex, shown in figures 4H to 4I. Continuing with figure 4, there were contributions from the ipsilateral and contralateral hippocampi, shown from figures 4J to 4O, the ipsilateral thalamus, 4P to 4R, and quantitative analysis in S&T showed that brain regions and thalamic nuclei that contributed to these monosynaptic connections within the organoid grafts. Notably, the visual cortex emerged as a prominent source of afferents in image S, while the lateral geniculate nucleus is notable in image T, underscoring the connectivity between the organoid graft and the host visual network. These findings shed light on the complex connectivity patterns of organoid grafts, although some questions still remain regarding potential pruning mechanisms as well as the duration of the analysis, which only looked at up to 3 months post-transplant. Figure 4, however, reveals the ability of the grafts to receive inputs from both adjacent and distant brain regions, again with a notable emphasis on the visual cortex and visual thalamic nuclei. Figure 5, the focus shifts towards exploring the connectivity between the organoid grafts and upstream sources of visual information, particularly the retina, using a modified herpes simplex virus system this time for polysynaptic anterograde tracing. As shown in figure A, 2 months after transplanting the non-fluorescent organoids, HSV GFP was injected into the contralateral eye of the host animal. A low micrograph image in B of the right hemisphere reveals the presence of GFP-positive cells within the organoid, indicated by the white arrow, and throughout various regions of the host brain, with the highest density of GFP-positive cells located in the entorhinal cortex, shown through the asterisks. To distinguish the non-fluorescent graft, human antigen marker STEM121, as before, was employed in image C. A high magnification view of the organoid in image D then showcases a dense population of GFP-positive cells that again co-localize with STEM121, showing robust HSV labeling. Importantly, these HSV-labeled cells that are GFP-positive within the organoid grafts exhibit neuronal characteristics, shown through their co-labeling with the neuronal marker beta-3 tubulin in image E. In contrast, these GFP-positive cells did not co-localize with GFAP in image F, suggesting that they're unlikely to be astrocytes. Continuing in figure 5, figures G to K confirm the presence of GFP-positive cells in the expected components of the visual pathway, including the visual cortex adjacent to the organoid, the lateral geniculate nucleus of the thalamus, and the optic nerve, specifically contralaterally, but not ipsilaterally, to the organoid graft. Quantitative analysis, images L and M, reveal significantly higher density of GFP-positive cells in the organoid compared to the adjacent visual cortex and visual cortex in naive animals without organoid grafts. This observation may reflect an overabundance of connectivity within the organoid prior to the occurrence of pruning. However, these findings unequivocally demonstrate that retinal output reaches the organoid graft through synaptic connections, elucidating the functional integration of these organoids into the visual cortex. Now, for the last figure of the paper, figure 6, the focus shifts to the functional integration of the transplanted organoids. So neural activity from the naive visual cortex and an organoid graft at 2 months post-transplantation are compared on the left and right sides, respectively. Image A shows the visual stimulation and electrophysiological recordings that were done. Representative single-unit waveforms from the visual cortex of a naive animal in B and the organoid graft in C are displayed, with gray lines representing the individual spikes and the black line indicating the average waveform across all of the recorded spikes. Raster plots of single units from the naive and organoid graft are shown in images D and E across 140 and 120 trials respectively, with the yellow line denoting the timing of a 0.5 Hz flashing screen stimulus. The same data is presented as peristimulus time histograms and event-related potentials, or ERPs, for representative evoked units from the visual cortex in the naive and organoid graft. Visual stimulation of the host animal also evoked ERPs in the organoid, albeit small in amplitude compared to those observed in the visual cortex of naive animals. Rose plots in images H to K show the number of times a single unit fired based on the orientation of a presented drifting grating from the naive and organoid graft. These units exhibited preferential firing for drifting gratings in a particular orientation. Black lines indicate the average number of spikes across trials, and gray lines correspond to one standard deviation above and below the mean. Angle measures around the periphery of the rose plot indicate the orientation of drifting grating presentation or degrees, while the radial axis indicates the firing rate of the neuron in response to that orientation of drifting grating. So this analysis demonstrates the capability of transplanted organoids to integrate functionally with the host visual system, exhibiting evoked unit activity as well as displaying feature cell activity. In conclusion, the paper came full circle to its original key points and its highlights, and showed that 1. Human brain organoids can integrate structurally and functionally with the injured adult rat brain. 2. Organoid grafts connect synaptically with the rat brain and can adopt the function of the visual cortex. And 3. Brain organoid transplantation has great potential as a therapeutic strategy for restoring cortical function. Thank you so much for listening to my presentation on cortical brain organoids transplanted in the injured adult rat visual system. And please reach out if you have any questions.